Okay, so we finished up last time by talking about the oral cavity and pharynx and esophagus and the process of swallowing. So now we're moving on to the next portion of the GI tract, and that is the stomach. This is a very large chamber in the GI tract that's main function is to really begin to break down materials as well as to mix them together. The stomach is composed of 4 different regions. You have the cardiac region and the fundus region at the top, the body, which is the main portion of the stomach, and then the pyloric region. And then you have 2 sphincters, which control materials moving into and out of the stomach. Those are the cardiac sphincter and the pyloric sphincter. So we have the cardiac portion right here, which enters from the esophagus. We have the top portion right here, which is the fundus. The body is the main portion of the stomach. And then we have the pylorus, which is the near exit portion of the stomach. Then we have your 2 sphincters, your cardiac sphincter right here, which separates the esophagus from the rest of the stomach. This makes sure that when food comes from the esophagus into the stomach, it doesn't go back up, as well as any stomach acid or any other materials from getting back into the upper part of the GI tract. And then on the other end of the stomach, we have the pyloric sphincter. And this, when the stomach is ready to move contents from the stomach into the small intestine, the sphincter will open, allowing for food to move through. And much like all of the other layers, or organs of the digestive system, you have all of your different layers, like your serosa, your muscularis, your submucosa, and your mucosa. So like I just mentioned, you have those 4 layers: serosa, muscularis, submucosa, mucosa. The one interesting difference that the stomach has, the other sections of the GI tract don't, is that your muscularis has 3 layers instead of the usual 2. So normally we have the longitudinal and the circular. Your stomach has an extra inner oblique section of the muscularis, which helps with mixing the contents. Additionally, your stomach has special structures in the tissue called rugae. These are little folds that remain relaxed when empty, but when they are filled with food, such as after you consume a meal, they start to stretch. So these rugae exist in the mucosa and the submucosa, and the reason that you have them is that the stomach can expand when you have food. It allows the stomach to actually stretch and get bigger. Then within the stomach, you have what's called chyme. This is what happens when the bolus, which is that crushed up food mass as it moves from the oral cavity to the pharynx to the esophagus, when it gets to the stomach, it starts to become more broken down and more pasty, kind of thick and lumpy and pasty, kind of gross to describe. But that is called chyme. That's because the food is being mixed with a bunch of digestive juices as it's being broken down. So then going a little deeper with the histology level, in the actual tissue, we have what are called your gastric pits. And these are opening sections of the tissue that have what are called gastric glands. The gastric glands are responsible for producing a bunch of different things like stomach acid and mucus and different hormones. And within the pits, you have a bunch of different cell types. So your surface mucus cells and your mucus cells produce mucus. They help protect the stomach from the actual digestive acid so it doesn't actually eat itself away. You have what are called parietal cells. These produce your hydrochloric acid, which sanitize the cell or sanitize the stomach, as well as convert this compound called pepsinogen to pepsin, which helps break down proteins. Your parietal cells also make what's called intrinsic factor. And your intrinsic factor helps with the absorption of vitamin B12, a very key vitamin. Then your chief cells produce pepsinogen. As I mentioned with the hydrochloric acid, your pepsinogen has to be converted to pepsin in order to break down proteins. In this pepsinogen state, it's inactive. So it has to work together with hydrochloric acid to be able to break down proteins. Then lastly, you have your endocrine cells, which produce all sorts of different types of hormones. So your histamine and your gastrin stimulate additional acid secretion, some more of that hydrochloric acid. And then you have another hormone called somatostatin, And this is basically an inhibitor for gastrin as well as insulin, which we're not really going to get into insulin right now. But what you need to know is that somatostatin is going to inhibit that stomach acid secretion by blocking gastrin. So, in terms of the actual way that the stomach regulates its secretions, there are three phases. There's a cephalic phase, a gastric phase, and an intestinal phase. So to start with the cephalic phase, this is basically initiated by any sort of interaction with food that's not directly putting food into the stomach. So tasting food on your tongue, smelling the aroma of food, just thinking about food, like thinking about your lunch or your dinner you're really looking forward to, that's going to stimulate the medulla oblongata brain to then set an action potential down to the stomach. Once that action potential reaches the stomach, this is where your enteric nervous system is going to become activated. That's that local nervous system that only the digestive system has. Once that gets activated, that's going to stimulate the secretion of your hydrochloric acid, which is responsible for sanitizing and keeping microorganisms away or destroy them in the stomach. And it's going to produce pepsin, which helps break down those proteins. It's also going to produce your gastrin and your histamine. Your gastrin and your histamine are hormones, so they are long-distance chemical messengers. So they're going to actually leave the stomach and go through the blood vessels all the way to back to the stomach, which is then going to stimulate further secretions. So it's going to stimulate more hydrochloric acid and more pepsin to be released. So think of this cephalic phase as kind of like turning on the car. We're getting things started. We're getting things moving because the body is anticipating getting food very soon. So we need to prepare for it. Next, you have your gastric phase. And this pretty much works exactly the same as the cephalic phase, except with the caveat that instead of the brain being stimulated by food, having thoughts about food or smelling food, in this case, we literally have food in the stomach, which is causing this pathway to occur. So instead of there being, again, stimulation by thoughts of food or interactions with food aromas, the stomach is going to be distended because that rugae is filling up with food content, stretching the stomach, creating a signal to then send to the brain. Brain is going to send a signal back to the stomach saying, hey, we have food in the stomach. So we need to produce more compounds to be able to digest this and break this down. So again, that histamine and that gastrin are going to be released from the stomach. And those are those chemical messengers that are going to go through the bloodstream and the back of the stomach and continue to produce more hydrochloric acid and more pepsin on top of what's already being produced. So in terms of the really basic way of thinking about it, think of the gastric phase as really hitting the gas pedal. So we have food that we have to deal with now. We really want to crank things up. We really want to step in the gas and get things going even faster. Lastly, we have your intestinal phase. So this is sort of the pump the brakes. We are all done with digesting and dealing with contents of the stomach, so we don't need to have these secretions and have all these pathways going. So the way this is triggered is that chyme is the pasty, partially broken down food mass. When that moves from the stomach into your small intestine, specifically the duodenum, that's the first section of your small intestine, either that chyme content is going to have a lot of lipids in it, or it's going to be very acidic from that stomach acid content. Either way, it's going to basically signal the brain, again, the medulla oblongata, which is then going to signal the stomach to stop secreting pepsin and hydrochloric acid. So we don't need this anymore because we don't need to break down contents. Additionally, you're going to have local reflexes. So the fact that the stomach isn't being distended anymore, it's not being stretched out because the food contents are leaving, meaning we don't need to produce those compounds anymore. Then lastly, hormonally, you have two hormones called secretin and cholecystokinin. These are two hormones that are going to go to the bloodstream. They're going to end up back at the stomach, and they're going to inhibit secretions as well. So they're going to block things like gastrin, histamine from producing more hydrochloric acid and more pepsin. So three different ways that you sort of pump the brakes for the stomach. Then within the stomach itself, when we have food being broken down, the stomach is also moving the food around. while breaking it down. So when we have this chyme, this again pasty, partially broken down food mass, the esophageal and the pyloric sphincter are closed because we don't want food getting back into the esophagus, and we're not ready yet for food to move into the duodenum, the small intestine, for the next phase of digestion. So those are closed. While those are closed, we're having two types of waves. We have your mixing waves, which is sort of this back and forth sloshing, just kind of moving the chyme around, getting it sort of evenly mixed, for lack of a better word. And then we have your peristaltic waves. These are the waves that are ultimately contracting to push the chyme content further along the GI tract. So when we've mixed up the food enough and we've broken it down, those peristaltic waves can force some of the food through the pyloric sphincter that's relaxing into the duodenum of the small intestine to continue on with digestion. Speaking of which, the next section we're going to talk about is the small intestine. So while we typically associate the stomach as being the big digestion site, the small intestine is actually the site of the GI tract where we have the greatest amount of digestion, largely because of the amount of enzymes we have breaking everything down in this area. Additionally, it's also the site where everything's going to be absorbed. We're going to get all of those important nutrients into the bloodstream or into the lymphatic vessels to be distributed about the body. The small intestine is broken down into three sections. The duodenum, which is the very small starting section of the stomach, starting section of the small intestine. The jejunum, which is the next section, and then the ileum. In your first two sections, the duodenum and the jejunum are really where we have the nutrient absorption. The ileum is really just continuing to pass along that food until we get to the large intestine. So structures we have in the duodenum, first and foremost, we have what's called the major and minor duodenal papilla. These are basically openings that come from the bile ducts and the pancreatic ducts, allowing for secretions to get into the small intestine to allow for the breakdown of food. We'll get a lot more into the liver and the pancreas in future slides. But this opening is where all those enzymes are going to be secreted. Most of them are going to come through the major, and then a few of them are going to come through the minor. Then we have three different structures or layouts that help increase the surface area of the duodenum for increasing absorption. We have what are called your circular folds, and these are in the mucosa layer and the submucosa layer. We have what are called villi. These are projections that we have coming off of the mucosa. And then within the villi, what I'm circling right now, we have what are called your microvilli. These are extensions or little sort of hairs coming off of your absorptive cells, which are on the villi. The absorptive cells kind of make what's called a brush border. These are basically just a group of hairs that are helping to catch the food particles and bring them into the cells so they can be absorbed. So, as I said, the important purpose of circular folds, villi, and microvilli are to increase surface area. So we want to increase that surface area so we can increase the amount of space that materials can be absorbed through, because we want to get as much nutrients absorbed in this area as possible. Then within the duodenum, we have all the different types of cells. We have absorptive cells, which, as the name implies, help absorb the digested food. They also do produce some digestive enzymes to break down different food contents. We have your goblet cells, which help produce mucus, as they do similarly in the respiratory system, in terms of the mucus production. Then we have granular cells, which help protect us against bacteria and these sort of microorganisms we don't want there. And then we have, of course, endocrine cells for producing those hormones that we always need. You have two types of glands within duodenum. You have what's called the intestinal glands, which sometimes they're referred to as the crypts of Leverkuhn. This is where all of your cells originate. So your absorptive cells, your goblet cells, your granular cells, and your endocrine cells all start in the intestinal glands. The absorptive glands and the goblet cells actually move out of these areas to the top of the villi, while your granular cells and your endocrine cells remain within the actual intestinal glands. So on this diagram to the right, where it says letter C, the intestinal gland is at the bottom. It's sort of this little pouch between the villi. That's, again, where all your cells originate. And then you have what's called your duodenal gland. And these are basically mucus glands that are in the submucosa below the mucosa. So again, on this diagram over here, where you see letter C, the duodenal gland is this little mass that lies beneath the surface. That, again, can allow for mucus secretions to come into the mucosa layer. So next part of the small intestines are the jejunum and ileum. And as we move from the jejunum to the ileum, gradually we have a decrease in the diameter of the actual tube that the food's moving through. The wall isn't as thick. We don't have as many of those circular folds and villi, so we're not having as much surface area. Thus, because of this, we don't have as much absorption as we move from the jejunum to the ileum. The duodenum and the first part of the jejunum are where a lot of absorption is going on. But again, as we continue down, there's less and less of these structures to increase surface area, so we're not absorbing as much nutrients in these areas. Within the ileum, you have a couple of special structures. First, you have what's called Peyer's patches. These are little lymphatic nodules that reside in the mucosa and the submucosa. And they can initiate immune responses against any sort of microorganisms that may be in the small intestine that we need to get rid of. Then you have what's called the ileocecal junction. And this is just the area where the ileum actually attaches to the large intestine. And then there are two structures, so food can't move backward. We have the ileocecal sphincter, which obviously, if we don't want things to move, will remain shut. And if it's open, will allow contents to move to the large intestine. And then, similar to your veins, we have one-way valves called the ileocecal valve, which just lets the contents move into the large intestine, but not back into the small intestine. So we always want the flow to be one way when we're getting to this point in the GI tract. So the small intestine has a lot of different secretions and things that it produces. The first of which I mentioned a few times already, is mucus. This does a couple things. One, it keeps the digestive tract lined so things can slide through, but it also protects against any sort of digestive enzymes or stomach acids. And there are a lot of acidic contents in the small intestine, both from the stomach itself as well as the pancreas. Then we have the digestive enzymes themselves. Examples of this are your disaccharadases, and these break down your disaccharides to your monosaccharides. So those are two types of carbohydrates. Disaccharides are a monosaccharide and a monosaccharide. If we break that down, it's just two monosaccharides. Then we have peptidases, which break your peptide bonds, which hold amino acid chains together. So we can break those peptides down into individual amino acids. And then movement, we have your segmental contraction. So that's helping to mix and evenly coat and increase the surface area of the food content, so more of it can be absorbed. And then we have your peristalsis, which is propelling that food mass, or that chyme at this point, down the small intestine tract. Okay, so before we go to the large intestine, we have a couple accessory structures that we need to cover that help with digestion. The first one's the liver. So the liver is basically this giant lobe mass that we have near the small intestine that secretes bile. It's made of 4 lobes. So you have a right lobe and a left lobe, as well as a quadrate lobe and a caudate lobe. Those are the minor lobes. You have a section that's referred to as the porta, and this is the entry point for all of your different blood vessels, any sort of ducts, any nerve or nervous tissue, sort of the enter and the exit point for the liver. And then you have a bunch of different ducts that run throughout the liver themselves. So you have what's called the right hepatic and the left hepatic duct. The right hepatic and the left hepatic duct combine to form what's called the common hepatic duct. Then you have what's called the cystic duct, which comes off the gallbladder. And then when we have the common hepatic duct and the cystic duct combined, those form what's called the common bile duct. The common bile duct is what actually empties the bile contents into the small intestine. So a lot of ducts. So to show you how all those connect together, in the liver, we have the right and left hepatic ducts. These come from the liver tissue, so they're carrying fresh bile. When those combine, they form the common hepatic duct. Then you have the gallbladder, which is attached to the liver for storing bile. That has the cystic duct coming off of it. When that cystic duct meets the common hepatic duct, that then forms a common bile duct. The common bile duct then runs all the way until it hits the small intestine, and it has a special structure called the hepatopancreatic ampullae. This is basically where the common bile duct is meeting the pancreatic duct. So your pancreatic duct is right here. We'll get to the pancreas in a second. It's going to meet the common bile duct at this key point. That's the hepatopancreatic ampullae. The actual contents are going to be delivered through the major duodenal papilla. That's what I'm circling here at 4. So in terms of the actual histology of the liver, we have those lobes, but when we further get into the microscopic level, the liver is made up of these structures called lobules. So the lobules are these hexagonal shapes that I'm drawing my mouse around. Each hexagon is a lobule. And then we have what's called a portal triad at each corner of the hexagon. And the portal triad contains 3 pieces: a hepatic duct for bile, the hepatic portal vein, and then the hepatic artery. At the center of each hexagon, you have a central vein. This is ultimately what's going to lead to the inferior vena cava, carrying blood back to the heart. You then have what are called hepatic cords. These are these white pieces that are radiating out from the center. These are composed of these smaller hexagons, so hexagons within hexagons. These are called hepatocytes. Your hepatocytes are responsible for actually creating the bile that the liver is going to produce and put in the small intestine. So hepatocytes make the bile. Then you have these other little canals called hepatic sinusoids, what I'm drawing my mouse over right here. They contain hepatic phagocytic cells, which help with an immune response, get rid of microorganisms we don't want. And then lastly, we have the bile canaliculus. And these are these little small bile channels that run along the hepatocytes. So the bile they produce then goes into these canaliculi, which will eventually go to the hepatic ducts to be released from the liver. So a lot of structures there. So the liver itself does many, many things. First and foremost, as I mentioned, is that it produces bile in order for digestion to occur as well as to be excreted from the body. The bile itself is able to do 2 things. It's able to neutralize and dilute the stomach acid, so it doesn't actually harm the body. And the bile salts, which are contained within the bile, are able to do what's called emulsifying lipids. Emulsification basically means when we take a really big chunk of lipids, so a really big fatty amount of tissue, and we break it down into smaller fatty droplets. When we have these smaller fatty droplets, they're small enough to be reabsorbed in the ileum. Next is the storage of nutrients in the liver. The big one is glycogen. When we have any sort of glucose that we aren't using or the body needs to store, it's stored as glycogen in the liver. The liver can also store things like fats, vitamins, and minerals, but for the most part, it's glycogen. Additionally, it's able to process a couple different nutrients. So it can process amino acids to make ATP, that's the energy compound. It also is able to activate vitamin D. So vitamin D is actually in an inactive form when it enters the body. It has to be activated, and the liver is able to do so. Vitamin D is responsible for maintaining your blood calcium levels. Additionally, the liver is involved with detoxification. So those hepatocytes that make bile are also able to remove ammonia, which is needed to be converted to urea to be excreted through the urinary system. Then lastly, your liver helps with synthesizing a lot of your blood proteins. So things like your albumins, your globulins, your fibrinogens, most of which you remember being helping with the blood clotting process from the very first chapter we covered. Okay, so in terms of controlling the biosecretion and release, this again has similar pathways to what we looked at in the stomach. So the vagus nerve is able to send an action potential to the liver. And ultimately, that is going to stimulate the liver to produce bile. Bile is going to go to the duodenum through all of those ducts and help neutralize stomach acid and emulsify lipids. Those bile salts are going to stimulate the release of additional bile from the liver. Additionally, you have two hormones, secretin and then I always mess up the pronunciation of this. Cholecystokinin, it's a mouthful, are going to be released from the duodenum, go to the bloodstream, and then return to the liver to produce more bile. So secretin and cholecystokinin are also released from the duodenum, which stimulate more bile production. Then we have the gallbladder. So I already mentioned this briefly when we talked about the liver, but the gallbladder is just a bile storage center. It's just a sac that sits on the portion of the liver just to store bile until the body needs it. Much like the liver, it is also stimulated by the hormone cholecystokinin and vagal stimulation from the brain. Ultimately, that bile is going to exit through the cystic duct and then join the common bile duct to enter the duodenum. And occasionally, there can be a blockage in the gallbladder through what's called gallstones within the cystic duct. Okay. Next is the pancreas. So the pancreas is a mix of endocrine tissue and exocrine tissue. So endocrine tissue produces your hormones, things that have to go through the bloodstream because they're long distance chemical signalers. Exocrine tissue is able to produce compounds that just get secreted into the tissue they're next to. So endocrine has to enter the bloodstream, exocrine just gets secreted directly into the target tissue. The pancreas is made up of three portions. You have a head, a body, and a tail. And then the endocrine and exocrine portions have special structures. So the endocrine portion that produces your hormones have what's called pancreatic islets. And these produce hormones like your insulin, your glucose, and your somatostatin. And then your exocrine tissue has these sections called acini, which are the same similar acini you saw with the salivary glands, same exact idea. And together they all form lobules. When the exocrine secretes some sort of product, it's going to go through a bunch of different ducts. So it's going to start at the intercalated duct, go to the intralobular duct, go to the interlobular duct, and then end up at the pancreatic duct. The pancreatic duct is then going to join the common bile duct, that special structure called the hepatopancreatic ampullae, which then goes through the major duodenal papilla into the small intestine. So to show you what those structures look like, your pancreas is hanging off of the duodenum of the small intestine. We have the head, the body, and then the tail of the pancreas. And then we have the pancreatic duct, which is this main vessel that runs all the way through and joins the major duodenal papilla. Then zooming in, we have a lobule, which is made up of your endocrine tissue as well as your exocrine tissue. Your endocrine tissue is responsible for producing all those hormones, so insulin, glucagon, as well as somatostatin. And then the acinar cells, so these little sacs right here, are able to secrete all the enzymes from the pancreas. Those secretions then start through the intercalated duct. They reach the intralobular duct. They then go to the interlobular duct, and then join the pancreatic duct. Pancreatic duct then joins the hepatopancreatic ampullae, again to be secreted into the small intestine through the major duodenal papilla. A little bit of the secretions will actually go to the minor duodenal papilla from the pancreas, but for the majority, it's the major. So, in terms of the exocrine tissue, the secretions from the pancreas are collectively called pancreatic juice. And this is basically just made of an aqueous component and an enzymatic component. So the aqueous component, as the name suggests, has mostly water in it, but it also has a couple of different enzymes like your sodium and your potassium, as well as your bicarbonate ion. Then the enzymatic portion has a bunch of different enzymes, which are help with breakdowns of a lot of different macromolecules. So your trypsin, your chymotrypsin, and your carboxyl peptidase all break down proteins. So they break down peptide chains into amino acids to be absorbed. Your pancreatic amylase breaks down polysaccharides. So we want to break down polysaccharides into disaccharides and then monosaccharides to be absorbed. So we're aiming to get something from like a starch to glucose. And then your pancreatic lipase breaks down your lipids. And then a special two types of enzymes. We have deoxyribonuclease and ribonucleases, and those break down DNA and RNA respectively. So lastly, for the pancreas, much like everything is regulated in a similar pathway between stimulation from the nervous system as well as hormonal control. So for the pancreas, that vagus nerve in the brain is going to set an action potential to the pancreas, which is going to stimulate the secretion of those enzyme-rich pancreatic juices from the exocrine portion of the pancreas. Additionally, you're going to have secretin that comes from the duodenum and cholecystokinin from the duodenum, which are going to both go to the bloodstream and then end up back at the pancreas, and that's going to stimulate the additional production of pancreatic juices. So stimulation from the brain, and then stimulation from the hormone secretin and cholecystokinin.